Good afternoon, everyone. Thanks for taking the time to listen about Gelmedix. Gelmedix is innovating the next generation of ocular and regenerative therapies using our proprietary and tunable hydrogel formulations, which is capable of delivering a variety of therapeutics, but is particularly well suited to deliver stem cell-based therapies. Our lead product is an integrated hydrogel and retinal pigment epithelial cell therapy to treat age-related macular degeneration and geographic atrophy. We've assembled a team with experience to move this project forward. We're currently focused on generating in vivo data for the retinal cell therapy application, and we'll be raising Series A financing in early 2023. Existing clinical data for retinal cell therapy has been promising, both from safety and efficacy signals. However, there have been challenges with both approaches. From a free cell injection aspect, some of the challenges are cell stress during injection, migration of the cells from the target area, aggregation of those cells, and that results in an inconsistent cell distribution. And the other approach is a preformed or solid scaffold, and that attempts to pre-create the desired cell monolayer. However, those scaffolds are often stiff, sometimes even non-degradable, have tissue integration challenges, and require a more invasive surgery. With the Gelmedix formulation, we look to address some of those major challenges with those approaches. What we do is we combine the retinal pigmented epithelial cells with our hydrogel precursors, injecting them with a standard subretinal injection. The Gelmedix formulation protects the cells during injection. It localizes the cells at the target tissue, minimizing aggregation and facilitating monolayer formation. Simply put, on the application, it would be a standard subretinal liquid injection. Once the material is deposited subretinally, in this case the retina is the yellow layer, the existing vitrectomy light is focused on the material, cross-linking the material into a soft solid hydrogel and fixing the cells in place. Degradation of the hydrogel is optimized for monolayer formation and tissue integration. Just a minute on the gel. The hydrogel is an extremely efficient photocurable hydrogel platform. As I mentioned, it can deliver a wide range of therapeutics, but in particular, it's enabling for vision restoring cell therapy. And that's because it has some natural constituents to it that are favorable to cells and really aid in tissue regeneration. Additionally, the platform is very tunable, from the variety of therapeutics to properties such as mechanical, biodegradation of the hydrogel, therapeutic tissue, therapeutic drug release, as well as bioadhesion, really making it a very versatile material. And so, in general, one thing I would say is the Gelmedix formulation creates more viable cells and monolayer formation. And in this example here, with the green calcine stained images, the top Gelmedix image at 7 days shows a very nicely formed mosaic and actually pigmentation as well. And the gel is gone at this point. And what we see with the free cell suspension is we're seeing some cell differentiation, evidence of that, with maybe a fibroblast cell that's shown in the center of the image. And to quantify that benefit to the cells, the Gelmedix formulation is able to deliver over twice the cell density at day 7 than a free cell injection. And again, this experiment was done in vitro using a standard Med1 subretinal needle. Sort of the robustness of that monolayer formed with the Gelmedix formulation is further shown by the 30-day post-injection staining, showing a nice retinal pigment epithelial monolayer and it maturing nicely. What we've done recently is advance the program into a large animal model. And in this case, again, comparing delivery of RPE cells via a liquid injection using the Gelmedix hydrogel precursors compared to a standard saline injection. The top images in this case are the bleb formations at day 0, just to give an indication of that. And then more interestingly, with some preliminary day 14 data, the bottom left shows, that's a free cell saline injection, and there's evidence of an epiretinal membrane. And looking back at day 0 imaging, it's not fully surprising when you note cell reflux from the injection site during post-procedure. And an interesting data set from the Gelmedix side in the bottom right-hand corner, at day 14, the hydrogel is well integrated. We're seeing RPE formation signals such as early signs of pigmentation. And just to take a minute and summarizing what we've been up to and what we plan on executing at Gelmedix. We recently raised some seed extension financing with the focus on that RPE cell therapy in vivo study. We're expanding our scientific team and we look forward to finalizing our licensing option for the cell asset. We're going to take that RPE in vivo data and use that to support an early 2023 Series A financing of $25 million with a goal to achieve an IND clinical trial submission, finalize licensing of the cell asset, as well as GMP manufacturing readiness. And just to close on the team, we'll put together a team well suited and experienced to achieve our goals. It possesses significant ophthalmic, biopharma product development, and successful new company formation. We've also engaged with clinician advisors who are experts in cell delivery to help advise our development pathway. As I said, we look forward to developing an innovative retinal cell therapy to address age-related macular degeneration and geographic atrophy. We're also open to partnerships for numerous different indications in the eye and potentially outside the eye as well. Thank you very much.